thanks.
Is it too loud or is that good?
Is this good or too loud? A little loud? It's a little echoey. Huh? Better? That's okay? Good. All right, thanks. Okay, so I just want to sort of give you a quick tour of where we've been so far. So in the first lecture, we started with the fact that we have 5 different kinds of eye movements, right? And I talked about the fact that we have 6 muscles on each eye and the way that they're innervated by the motor neuron pools, the way that we need to drive those motor neurons based on basically the mechanical properties of the orbit and surrounding tissues and the muscles. And we came up with this idea of a pulse step that followed both a biomechanical analysis of the ocular motor plant and then was confirmed by single unit recordings in alert behaving monkeys. And then we switched onto the vestibular system, and the vestibular system actually generates one of those 5 types of eye movements I initially introduced, right? So you have voluntary eye movements like saccades, pursuit, and vergence, but then you have 2 classes of involuntary or reflexive eye movements. One is the vestibular ocular reflex, which I've discussed, and the other is the optokinetic reflex. So if you close your eyes, would the gain of the VOR go down, or would it stay where it is? What do you think? So if we we're checking your vestibular ocular reflex gain and we turned out the lights, would you expect your VOR gain to go down? You would. Let's say if you turned out the lights and you checked your VOR gain at 5 hertz, would you expect it to go down? You shouldn't, right? Because I showed you 2 plots where I showed you the VOR gain is very good at higher frequencies. It's only at very low frequencies that the VOR doesn't function very well. And that's sort of the exercise that you're going to go through, right? I showed you those Bode plots and showed you the mid-frequency range where we have high gain as far as endolymph movement for head movement. There's the low band, the low frequency band and the high frequency band. And the point is that reflex, without any visual input, keeps your gaze on target as you move your head over most of the range of movements we generate without any visual feedback, right? It's really at very low frequencies of movement that it doesn't work so well because of the biomechanics of the canal. And there actually vision helps, and that's the optokinetic reflex, right? And the optokinetic reflex is really bad beyond 0.1, 0.2, 0.3 hertz. It has a pretty poor gain, right? So the vestibular ocular reflex is driven by the vestibular system, not the visual system, right? So you have a vestibular motor pathway. Okay. And if we look, this is sort of relevant when you look at the exercise that you guys have, the homework, just to think about what that pathway is trying to model for you, right? What you're trying to measure is sort of a feed-forward pathway that connects the vestibular end organs to the eye muscles and understand how it's compensating for your gaze, right? Keep your gaze stable as you walk through the world, okay, without visual input. Okay, and then in the last couple lectures, I focused on understanding the coding at these different, so we can build mechanical models of how the canals work, for example, in the vestibular system. The question is what do the neurons actually encode, and are those biomechanics reflected in, can you make predictions, kind of like we did in the motor system for eye movements? And I made the argument that, in fact, you can. We have 2 kinds of afferents, regular and irregular afferents. And then I showed you this example of a step input. So a step input of velocity is really basically a DC input, right? It's as low as you can go in terms of frequency. And what you see there is no longer velocity coding, but this sort of transient response, right, in response to the initial acceleration. Then it dies out over constant velocity. And then at deceleration, when you stop moving, you see this sort of inverted response, okay? And that's what that second part of the assignment for the homework is going to make you look at, that step response. And I showed you a little video. I probably wasn't so clear, but the point I was trying to make with that video is if you think of a kid spinning around in the park, right, at a constant velocity, like you go out to the park and you spin around when you're little, and if you close your eyes, right, and then you stop and you open your eyes, you can make yourself fall over, right? You guys can all go do that on the main quad when you're done, right? Prove it to yourself. And that's basically taking advantage of the way these sensors act. So the idea is if you spin yourself around, eventually your afferents are not encoding that velocity anymore because of the biomechanics. You stop yourself, then you get that kind of acceleration, deceleration response. Even though you're not moving anymore, you get this false sense of motion because of the way the canals are organized, or their mechanics. When you open your eyes, you'll have this sort of beating movement of your eyes, and it makes you fall down, right? So that's what kids play with in the park. So we all know from very early on that we can kind of play with the biomechanics of our vestibular system. Okay, so we looked at afferents in other ways too. We didn't just look at their dynamics using sine waves and step inputs. We looked at things like neural detection thresholds, information coding, and we came up with the fact that there are two streams, right? And then what I started with last lecture was the fact that everything we talked about was really applying passive stimulation to the animal, right, or the person. But mostly we're generating our own active movement, and I explained why it's important to be able to dissociate motion that you experience because of things you've done versus things that are happening to you, right? We talked about this idea of postural control. You have pathways, 3-neuron pathway from the afferent central neuron to the motor neurons in your spinal cord that help you keep upright when you experience unexpected vestibular input. But if you actually have expected vestibular input, you don't want to mount postural responses. You want to actually make a movement. You don't want to generate a compensatory response that'll hold you stable in the world. And then I finished at the end of the lecture by talking about recordings from single afferents. So the same afferents we've already talked about, regular and irregular afferents. And the idea in the literature is that there's an efferent group of cells that projects back out to the canal, and that maybe during active movements this would change the coding range so you don't saturate your sensor, was the general idea. But if you actually look at neuronal responses, and this is about where I finished up. So this would be an active head movement made by a monkey, right? Making a head movement like this or a head movement like that. And you can see we can then play that same head movement into the turntable and rotate the animal exactly the same way. And what you can see in these two conditions is the neuron responds pretty much the same way. And you can fit a really simple model, which is some resting discharge, some gain times head velocity, because most of this is in the velocity coding range. Maybe a little bit of a lead, which is represented by this head acceleration term. And you can basically use the same model to explain the neuron's responses in both conditions. So they act like, basically a sensor, right? They just tell you head and space motion, regardless of how it's generated. And then I finished just showing this curve, which says your resting discharge doesn't change for basically passive versus active conditions, nor does your sensitivity to motion. So these are just each of these dots or triangles, or I guess diamonds, represents a different afferent, right? So you can measure the parameters I just showed you for each afferent, and then you can plot them in the two conditions. Okay. So the next thing you do, say, okay, we're going to now go in one more stage. So that's the nice thing about the vestibular system. It's a very simple system. We can go basically structure by structure and see how information is processed. So now we're going to just move in one more neuron. So the afferents, remember, both the regular and irregulars, terminate on these central neurons. And we're going to focus on the neurons, not the ones that generate the VOR, okay, but the neurons that are involved in posture. And that also send projections up to the thalamus. Okay. And if we do that same thing and we basically rotate the monkey, in a trajectory that is similar to what he would generate for an active head movement, the neurons are really nicely modulated. So this is just firing rate, right, as a function of time. And you can see they really follow very nicely the head movement that was applied. And then, voila, if you look in the active condition, right, here you see something quite different. So the monkey's now moving his head on his body actively, right? Remember, the afferents encoded these two types of stimuli exactly the same way. But one neuron in, you see this big difference where no longer is the neuron encoding head velocity. So that's a pretty big change, right? And the thing to bear in mind is these neurons are getting input from the afferents that are actually encoding that head motion, which suggests that something is canceling this response. And then you can look at a slightly more complicated situation where you've got the monkey on a turntable and you're passively rotating, but he actually moves his head on his body. Okay, so he's got a passive component of the head motion, which because we're moving him in space as experimenters. In addition, he can generate additional motion of his head on his body. So his head is moving through space because of our sinusoid that we're applying, in addition to his active head-on body movement. So the total head in space motion would be this trace here in black. This is the passive component, and this would be the active component. And if you look down here, this is just a similar for another example where the animal's moving so much he actually moves his torso actively. So the head is moving through space because he moves rotates his torso. And you see the same thing. And what's pretty, I think, fascinating, right, is that the neuron is just sculpting out that unexpected applied movement. It's totally ignoring, for this particular neuron, the self-generated component of the vestibular input. So if you record from an afferent, just to give you again something really clear, during this same protocol, it would basically follow a trajectory like that blue dashed line, right? Total head in space velocity. So here you have a selective encoding of vestibular X afferents, right? The applied vestibular stimulus. And it's actually ignoring the reafferents. Okay. So the question is, answer to the first question is, where does this occur? And we see this selective representation of unexpected or X afferents in the vestibular system at the first central neuron, right? And that's sort of a surprise. And it's a really clean computation. And you can see this, what I showed you is for rotations, for horizontal rotations that would activate the horizontal semicircular canal, but it's also true for translations. So if you make active translations, you would see the same thing. And it's common across species. So you see this in mice, you see it in rhesus monkeys, you see it in squirrel monkeys. Okay. So what I also just really emphasized, right, is the fact we know from other experiments that the neurons here, these VO neurons, are getting an afferent input, and that afferent input is encoding both X afferents and reafferents, right? So in order to just encode X afferents, we need some sort of cancellation signal, right? We need something that's going to subtract out the component of the movement that the monkey generated himself. Okay, so if you go back and think about those experiments that I just showed you, in one case you're rotating the monkey passively, right? So he's sitting in a monkey chair on a turntable. We rotate him back and forth. And in that case, you're activating the vestibular system. But if you think about the other experiment, there's some other things going on, right? So you can kind of come up with sort of a Boolean logic of, well, in this case, we have the vestibular system, but we also have proprioception, right? Because the monkey's making a neck movement, so he's stretching his proprioceptors in his neck. And you guys all know what proprioception is. The monkey's also generating a motor command to move his head, right? So there's some motor command that's going down to the muscles to make that head movement. The brain has access to basically the motor command information. And then you could argue there's some higher level knowledge that the monkey might have of the fact that he's making that head movement. So, maybe it's just that you have a proprioceptive signal that's canceling the vestibular input. Or maybe it's just that the motor command is canceling the vestibular input. So you can check those things. And the way that, you could probably think logically how you might do those experiments. So for example, to check whether or not there's a proprioceptive input on these neurons, what you can do is keep the head stationary relative to space. So there's no activation of the vestibular system, right? The only way you activate the vestibular system is moving the head relative to space. So you clamp it fixed relative to space, and then you rotate the animal's body underneath it. So it would be basically the body rotating under an earth-fixed head. And the neurons never respond to that particular stimulus, suggesting that they're not sensitive to proprioception. It's not that you just have a negative input from the proprioceptive system that can cancel things out during active movements. And the other thing, so this is all done passive. The other thing you can do is look at whether or not just having an efferents copy or a motor command present is enough to make an inhibition or cancellation signal that will cancel out the self-generated movement, right? That's sort of a logical thing to think about. Maybe you have a motor command that goes in and just shuts off the neurons in a way that's perfectly complementary to that signal that's coming in due to the active head movement. So to check that, what you can do is have the monkey making normal active head movements and then clamp his head using like an electromagnetic clutch. So he can't move his head, though he thinks he can. He tries to make a head movement. And then you measure the torque that the monkey's generating against the head restraint, right? So the torque is like a rotational force. And if the animal's trying to generate a torque, what that means is he's trying to generate a head movement, right? So we know that this is a motor command. So the presence of torque means the presence of a motor command. And the neurons don't actually respond to that. Okay. And that's a little different than some other systems where people have looked, sort of model systems, looked at reafferent suppression, like the electric fish, which has been a very nice model. In the electric fish, the mechanism seems a little different and will actually shows inhibitory low sort of responses in a condition like that, in the sort of electric fish equivalent of this circuit. Okay. So you can go through a bunch of, what if this and that, and what if this and that, and ultimately what it looks like is, in the literature, is if you have reafferent suppression, right, during active movements, the suppression of vestibular input, this occurs in conditions where the sensory feedback that the animal experiences matches that which was expected as matches that expected on the basis of the motor command that was generated. So the idea is that the monkey has been generating head movements all his life. His brain has some sort of internal model based on all that experience of when he moves his head, or she, that you have a certain expected sensory inflow, right? So there's an internal model of the expected consequences, expected sensory consequences of a motor command. So this would be the case during normal movements. The idea is that during normal movements, the model is appropriate, this internal model, to give you a good representation of what sort of sensory feedback you should expect, right? So what I just said should sound to people a bit like motor learning, right? Because the idea is in motor learning, the brain, in particular the cerebellum, computes an estimate or an internal model of the sensory consequences of an action, which it then compares to the actual sensory feedback, right? And the idea is that by comparing what you planned and what actually happened, you compute the difference, and that's called a sensory prediction error, right? And that sensory prediction error signal is then what is used to update the motor program as well as sort of fix your internal model so that it's correct, right, to keep it calibrated. So that's the sort of standard way of thinking about motor learning, but we're talking about something a little different here. We're talking about the selective encoding of sensory X afferent. So how does this pertain to this problem? And the ideas are actually kind of linked if you think about it. So in the present context, the computation of a sensory prediction error also enables the distinction between vestibular X afferents and reafferents, right? So if you're generating a movement and you have a match, you know it's reafferents, right? If you're experiencing some sort of vestibular inflow and it doesn't match what you expect, then it's probably X afferents. So by computing the difference between expected and actual sensory inflow, you can make the difference. Okay. So the question is, how is this actually computed in the brain? And as you might guess, we're going to go look at the cerebellum a little bit and use this as an example of computation for what the cerebellum does. So as already alluded to, there's evidence for the fact that the cerebellum might play a role in other suppression of reafferents, and sensory reafferents. One really beautiful body of work is in the electric fish. So there's electric fish swim around in the mud. They don't have great vision, and they basically generate these electric fields to know where their prey, their mates, the boundaries of their environment are. But they also will modulate these fields not only because of objects that are near them, but based on their own movements. And what they learn to do, or what the cerebellar equivalent in the fish learns to do, is dissociate what sort of modulation is due to the fish's own movement versus what modulation is due to the movement or presence of prey or mates or other interesting features of the animal's environment. Okay, so that's a really beautiful series of work by Curtis Bell, Nate Sawtell, who's now at Columbia, and colleagues, which has had a really been a great model system for looking at computations. There's also been some work looking at this sort of interesting question. I'll get to this at the very end, of why you can't tickle yourself, right? Most of us can't tickle ourselves. Are people familiar with this literature about tickling? Yeah, right? So Daniel Warpert's group looked at the fact that you can't tickle yourself, and the idea is that based on fMRI studies, he actually found that the cerebellum serves a similar role in the basis for this idea that you can't, the suppression of self-generated tactile stimulation versus externally applied. So the idea is you can go look at the vestibular cerebellum to try to understand what's going on here. In this case, so what you can do is take advantage of, again, the fact that this system is super simple compared to some of the other systems, right? So instead of recording from the vestibular nuclei, we're going to record now from a part of the cerebellum that sends direct projections to the vestibular nuclei, okay, called the rostral fastigial nucleus. And it also projects down to the spinal cord. If you lesion this area, we know you have problems with posture and balance. Okay? So it's very consistent with what I've already told you about these VO neurons in the vestibular nuclei that show this suppression. Okay, and these neurons also receive vestibular and proprioceptive inputs, which seems consistent with the sorts of roles that they would need to be doing. Okay, so have people seen a schematic like this for motor learning? Yeah, right. So this should look pretty familiar to you. So the idea is, you've got a motor controller. It generates a command. Things occur out, in the world. You have a sensory system that then senses, your proprioceptive movement, maybe the change in visual inflow, your vestibular input if you're walking or moving your head. And then you also have a forward model that estimates the sensory consequences of what that motor command should have elicited, and you compare your prediction with what actually happens and compute a sensory prediction error, right? So, and then you can use that error to either update your motor command, right, as well as update your forward model if you have a persistent error, right? So the idea is, if in the case of passive motion, the prediction is if you have a neuron whose responses are consistent with the computation of a sensory prediction error, you'd expect them to respond robustly, right? Because everything's unexpected, so there'd be no forward model or prediction because everything is passively or externally applied. Okay, that's what's sort of indicated over there. So then you can think of the other case, right, where you have instead active movements. And in active movements, basically, if neural responses are consistent with the computation of a sensory prediction error, you'd expect them to be unresponsive, right, during self-generated active movements, because the brain should have a forward model that is going to be able to predict the sensory consequences of that active movement. So you would expect a good match between the prediction and the actual feedback. So what you can do is, again, now go into a part of the cerebellum that seems logical. So again, this is the rostral fastigial nucleus, which is one of the deep cerebellar nuclei, and record and see if the neurons behave in a way that gives you any clue that a sensory prediction error is maybe being computed, right? Or some neural correlate of this sort of black box. Like, it's very easy to come up with black box diagrams. The question is, do they actually, and they, maybe they are logical, but do they actually map onto anything you can actually record in the brain? Okay. So the computation that we're looking for is something like this. This is just, putting in another kind of slightly simplified version. You have a forward model, right, which is the expected sensory feedback. You compare that to the actual sensory feedback, and then compute a sensory prediction error. So if you go into the fastigial nucleus, one of the really interesting things is there are two classes of neurons. There are neurons that are just responsive to vestibular input, and they're called unimodal neurons. And there's a second class of neurons that are sensitive to vestibular input and neck input. And they're responsive in such a way that they're perfectly complementary. And because of their complementary responses, they effectively end up encoding body and space motion, and I'll show that to you in a second. Okay. So this is the way you can test these, and these are protocols that you've already seen, right? So this is the standard whole body rotation, which only stimulates the vestibular system, right? So this is just vestibular stimulation. This is body under head rotation, so the body's fixed relative to the world. You stretch the neck under the body, so that's proprioceptive stimulation. And then this third, in this case it's passive, but you basically rotate the head on the body. And clinicians do this actually to test people's vestibular ocular reflex as well. Passive head on body rotation. Here you both have the head moving relative to the world, so vestibular plus you're stretching the neck, so proprioception. And if you look at these unimodal neurons, right, the head is moving in space here and the head is moving in space here, and they respond the same way. When you're stretching the proprioceptors, they don't respond at all. Okay, so they're really vestibular neurons. And they encode head and space velocity. And the bimodal neurons are a little different, as you might guess. Same three protocols. What you see is they only fire in the first two protocols. So they fire when you stimulate the vestibular system. They fire in a modulated way when you stimulate the proprioceptive system. And then when you stimulate both together, kind of the way you would during a normal head movement, but this is passively applied, they actually cancel each other out. So if you look across these two conditions, what these neurons are effectively doing is coding body and space motion. So it's kind of cool. There are two channels, one that encodes head and space motion and one that encodes body and space motion in the passive condition. Okay. So the next question is what happens in the active condition, right? Because that's what we're really interested in. So this is just a different way of showing you data like you've seen already for the vestibular nuclei neurons. These are passive movements and multiple trials of each below. So these are just spikes of, I don't know, maybe 10 trials. The gray here represents the average firing rate. The blue line is a good fit through it based on the actual applied head motion, right? And then these are active head movements. Why are they more variable than these? Well, because you can just apply the same movement over and over again when it's passive, but when it's active, there's always variability, right, in movements. So these are all pretty similar. The animal may be trying to do the same thing, but there's always a little bit of variability movement to movement to movement. But regardless, what you see down here is over the trials, that neuron is not responding very well, right, to active movement. The dotted line represents how much it should respond if it responded the same way it did during passive, and you can see that the neuron is pretty insensitive to active movement. So that's a unimodal neuron. You can do the same thing for a bimodal neuron. They're also not responsive. And then you can do the same thing where you do combined stimulation. You see they selectively encode the unexpected movement, right? The vestibular reafference. Okay. So we go back to this hypothesis, right, that we formalized with a sort of motor learning type model. And what you could think about how to test this sort of hypothesis more directly, right? So it's consistent with what we just saw for active versus passive in the cerebellum. So it looks like the cerebellum is doing sort of the same thing that we see in the vestibular nuclei. That's encouraging if we want to look for a cerebellar mechanism. But how do we really test this model? So the idea is if this is actually true, reafferent suppression should not occur when the relationship between the motor command that's generated and the actual sensory feedback that's experienced is altered, right? So how do you do that? Well, that again starts to sound a bit like motor learning. So what you can do is take a monkey, have him sitting, generating active head movements, but you can have him hooked up to a little torque motor so that when he tries to move his head, there's some resistive torque applied so he doesn't move his head as far as he thinks he's going to move his head. But in order to get his reward, he has to learn to move his head more, right? Or generate a bigger motor command to not move his head more, but generate a bigger motor command to move his head as much as he used to be able to. So this is like a motor learning protocol, right? So you have normal passive movements that you can apply, normal active movements, then you can put on that torque. So the monkey has to fight the torque to make the head movement. learns over many trials, and then once he's in that learned state, you can turn the torque off every trial, or a 10 trials or so, and look at catch trials and see that the monkey's going to overshoot, right? So that's what this looks like. So the idea is we have first two columns are just passive movements that are applied, active movements that the monkey generates, and then after you've got this sort of baseline characterization of a neuron done, you put on the torque. The monkey can only move his head about half as much initially because he wasn't expecting the torque. There it is. He has to deal with it. Takes him maybe 40 to 50 trials to start making head movements that look like his initial head movements. Right? So his goal is to get back to head movements that look like this, and by about 50 trials it happens. And then once you're in steady state in motor learning, you can take the torque off for these catch trials, and you can see the monkey overshoots, right? Because he's updated his motor command, right, to deal with this new environmental constraint. So what do the neurons do? So the first two rows are things you already know, right? The neurons respond to vestibular X afferents. They respond during passive stimulation, but they don't respond very well to vestibular reafferents when the monkey makes his own head movement. So then the sort of interesting thing, right, is when you turn, remember the model we're testing is we should be able to make reafferent suppression no longer occur if we alter the relationship between what the monkey does and what he expects, right? So if he makes a head movement but he doesn't get the sensory feedback he expects, then there's a mismatch. And that's what you see here. So right after we right after we put on the motor torque, and the monkey can only moves his head about half as much, you can see the neuron, even though the head movement's much smaller, is much more responsive, and its sensitivity in terms of spikes per second per degree per second of movement is the same as it is in the passive condition. And then you can see over time the head movements get bigger and bigger, but the response gets smaller and smaller, right? So what you can see is this sort of dynamic updating of the internal model by looking at this change in the neuronal response. And then the other thing that you can see is here we've got a pretty good sized head movement and no response. You throw in a catch trial, right, where the monkey then doesn't have to deal with the torque that he's now learned to expect. And you get this big overshoot, and the neuron follows that huge head movement, right, with the same sensitivity as passive. So these are big active movements, and the neurons are really responsive. So another way to look at this data, which might be helpful to think about, right, is just in terms of the normalized sensitivity relative to passive movement. So here's passive, here's active, so we have big suppression, just in the regular way people look at it. And then this is the first trials of the torque being on, so sensitivities are as if, even though they're active movements, as if the neurons are experiencing a passive movement or vestibular input. And then you see over time this reemergence of the reafferent suppression. And then again for catch trials you get this big response, which is right on the mark with the passive sensitivity. And a way to now directly relate this to error, or sensory prediction error, is you can compute the head velocity error of these movements relative to the monkey's goal, right, which are these active movements it made initially. And you see that very nice sort of symmetry, right, between the normalized sensitivity and the head velocity error, right? And you can look at this sort of the way people look at motor learning curves, trial by trial. And again, the peak head velocity and the neuronal sensitivity follow very similar relationships. Okay. So what I just showed you is a test of this hypothesis, right? That the idea is that if in fact there's an internal model involved that computes a sensory prediction error, we should be able to check it by altering the relationship between what the monkey's motor command what the monkey's learned to expect for a given motor command and what it actually experiences. The neural responses in these cerebellar neurons are consistent with this rapid updating of an internal model, right? So that just go back to this sort of figure, and I think this is an interesting point. If you look in the literature, there are two separate streams of work where people think about reafferent suppression and think about motor learning. And the idea is you can really use the same model to explain both, right? So sensory stimulation that's externally generated rather than self-generated, you can use this model to explain that, but you can also use it to explain changes that you would see in sort of cerebellar regions when you have a change in the effector or the world during some sort of motor performance. And I guess the general idea to think about is, you're using a system, this sort of motor learning system all the time, right? As you're walking down the street, if you start to, start to veer off the curve and you have to compensate, that's an unexpected vestibular perturbation, right? And this circuit can compute that for you on the fly and tell you what part of your movement's unexpected. But let's say you're doing something and every time you do it, you have the same persistent error, right? If you have an error that becomes persistent, then what you want to do is update your forward model, okay? So one time the forward model will tell you, did I expect this or didn't I expect this? And will help you sort out the difference. But a persistent error says you have a problem with the model you need to recalibrate. Okay? Okay, so what's the functional significance? I already mentioned this at the beginning of the lecture, that we can use this system to determine whether we expected to do something here. If you generate a purposeful movement, you don't actually want to have the vestibular spinal pathways sending compensatory commands down to your motor neurons to generate a reflex, right? So it's good to turn that pathway off when you generate your own movement. But let's say you have some unexpected perturbation. You want nice modulation in that pathway, right? Because you want to be able to send the proper commands down to your spinal cord to keep yourself upright. Okay. So you can just go and test and see, do the vestibular nuclei show these same responses that I just showed you? And I can tell you, if I went back and recorded from the vestibular nuclei, they would pretty much look exactly like what's shown here. They really inherit this same behavior, so they're really consistent with the idea that you're basically doing this computation online in a way that's appropriate for calibrating the motor circuits. Okay. Okay, so when I introduced the idea of why do you want to be able to dissociate self-generated versus externally applied sensory input, I talked about the fact for the vestibular system, it's important for calibration of behaviorally relevant modulation of the vestibular spinal reflexes, and also, basically using them in a task-dependent way. Using them only when you really need to compensate for something that's unexpected. But I also mentioned initially that a reason we need to do this is perceptual stability. I made this analogy of the fact that if anyone who's experienced an earthquake, it's a pretty profound sensation, right? That wow, I didn't just move, what was that, right? So how is the brain doing that? And what you can do is look at ascending pathways, which I'll have a little bit of time to talk about a little bit more. So the vestibular afferents are down here, the vestibular nuclei here. We focused a lot on the cerebellum, right, which is over here, and the RFN. But the vestibular nuclei, in addition to sending projections down to the spinal cord and getting input from the RFN, right, which is part of the cerebellum, send direct projections up to part of the thalamus. So you guys, I'm sure, have heard about the thalamus in terms of the visual system and the auditory system. And in the visual and auditory system, there are discrete nuclei, right? So the dorsolateral geniculate and the medial geniculate that process vestibular and auditory information respectively, right? But in the vestibular system, there's no dedicated nucleus. Instead, we have a posterior pathway that goes through what's called the VPL, the ventral posterior lateral thalamus. If you look in a textbook, that's really a somatosensory nucleus. So if you go and record from neurons in that structure, they all, or most of them, respond to somatosensory input. In addition, many of those neurons are sensitive to vestibular input. And some of them are also sensitive to proprioceptive input. And this is a place where you start to put together signals, as in the cerebellum, so that you're integrating multiple cues about your self-motion, right? So you can keep track of how your head's moving in space, how your body's moving in space, and start to put things together to keep track of the movement of different parts of your body as you navigate through your world. So most of the work done to date is really, again, sort of standard, apply a passive stimulation, kind of like a standard engineering approach, record the neuron responses, look for nonlinearities, look for compute Bode plots. But you can also, of course, record from neurons during unexpected versus self-generated movements. And as you might not surprise you, since the vestibular-only neurons are the ones that project up to the thalamus, these neurons actually only encode, or robustly encode, externally applied movement. So already at the sort of gateway to the cortex, you see that you have basically a selective line for encoding unexpected self-motion. Okay. So the idea is that this pathway is, if you actually measure the sensitivity, even more selective than the neurons below. So these neurons are like 90% sensitive to, or 90% attenuated to active movements. So the idea again is that these neurons are very important for the perceptual stability. Again, am I moving, or did somebody move me? You can tell that if you go and basically record from one of these ascending thalamic neurons. So I think most of you have already seen this work, so I just put this up again to make the link that this is actually a kind of a nice simple system, because we can actually look at, and one of the nice things about the vestibular system is we can look at motor pathways, right? We have a 3-neuron arc linking the vestibular afferents to spinal reflexes, so we know what those pathways are trying to do, and we understand the functional significance. Pathways like the somatosensory system are more complex, because the pathway between sensing something and action is not as direct, right? We're not going to be able to compress it down to a 3-neuron arc. But there's been some very nice work done by, again, Daniel Wolpert's lab, and I'll just tell you briefly, and people can go look up this paper, but it's a real classic if you haven't seen it, where you basically have a tickle robot that tickles the subject, right? So you have either the experimenter producing the stimulus to touch the participant's left hand, or the participant producing self-touch using his right hand. And what you can do with this tickle robot is basically add a delay between tickling the robot, or add a rotation between how you tickle the robot and how it tickles you. And with an increased delay, things get more and more tickly, and with an increased rotation, things feel more tickly. So again, it's this idea that you need this match between what your motor command is doing and what you experience. And if you have a mismatch, you actually let more information through. And I'll just finish with this. This is a paper that came out this past year by Nate Sawtell's lab in the auditory system, and it looks like there's pretty much a comparable circuit in the auditory system, and this I think was done in mouse, that cancels responses to self-generated vocalizations. Okay, so just like you have suppression of self-generated motion early in vestibular processing, this is very early in auditory, at the first central stage of auditory processing in the DCN, the dorsal cochlear nucleus. You also see suppression. Okay. I seem to have lost my cursor here. Oh, that's weird. I can't see the cursor on the. So is anyone an IBM person? I've had to switch to an IBM for these lectures, and I can't seem to get myself back out of this. Let me try this. Okay, that does it. Yeah, I think it's okay now. Yeah. Okay. Okay, sorry about that. So I'm just going to finish. I'm not going to get through this whole lecture, obviously, but I condensed it before I sent it in today. So what it's going to do is give you a more general view of higher level vestibular processing. Just to remind people, this is the cerebellum. Of course, we've got all the cerebral cortex. So this is a view of the cerebellum. And we really focused in that last lecture on just the vestigial nucleus. So recall, I think many of you probably know that there are 3 deep cerebellar nuclei that get projections from the cerebellar cortex, the Purkinje cells. And the most medial of them is the vestigial nucleus. And the vestigial nucleus is interesting. The more rostral part is vestibular. The more caudal part is actually eye movement related and seems to be involved in calibration of saccadic eye movements. And the vestibular nuclei is also, you can think of as a deep cerebellar nuclei because it does get some direct cerebellar input. All the colored areas on this show you areas that have neurons in them that if you rotate the animal in the dark are responsive, so they're vestibularly sensitive neurons. And for example, I'm going to focus now on one area that gets direct afferent input as well as vestibular nuclei neurons. So the vestibular system is unique across all sensory systems in that the afferents not only project to the vestibular nuclei, but they actually send some direct projections up to the cerebellar cortex. No other sensory system does that. And they also send some direct projections down to the deep cerebellar nuclei. Okay, so let's look at what happens with one of the functions of this area back here. This is called the nodules uvula. So it's lobules 9 and 10. So it's at the very back of the cerebellum. And you guys remember I kind of alluded to this earlier at the very beginning of my lecture. If we do a step input of head velocity and we record from an afferent, this is the response you get. And you can predict that from that second order differential equation that we came up with to explain the biomechanics of the end organ, right? And the idea is that, so this is, you can think of as a DC input in terms of velocity. So it's at the far left side of that Bode plot. So things are really in phase with acceleration. So acceleration is here and deacceleration is here. And you see this nice response that reflects that, right, in the actual single afferents. And again, there are 2 time constants governing that. So one would be super short, so you wouldn't really see it. And there's another one that's longer. And this is kind of relevant for the exercise you guys are going to go through. So this is just a schematized version of that. We have head velocity input. We have afferent firing rate. And then if we go, and the time constant is about 5 seconds, okay, of the longer of the 2 time constants for this. If you go into the vestibular nuclei and record, the time constant's almost 4 times longer, okay? And that suggests some sort of integration or memory to keep that signal around longer. And if you record people's either eye movement, so this is a schematic version of the VOR, right? And these little fast eye movements are the quick phases. So the slow phase is the compensatory part. You can only move your eyes so far, so it flicks back. The flicking back is the quick phase. So the slow phase component has the same time constant as a vestibular nuclei neuron. If we could measure somebody's perception, it would look similar. It would have that longer time constant. It's about 20 seconds. So what you can do, this is a lot of text, so I won't go through the whole text. You guys can read that. But the idea is that I showed you the nodules uvula. There are nice connections between the vestibular nuclei and the nodules uvula. It's basically a recurrent loop. And it looks a lot like that recurrent loop that I showed you for the prepositus. Remember I said the prepositus is the neural integrator. It takes a velocity signal during saccades, integrates it to make that position signal to hold your eye constant in the first lecture. So you can think of basically a loop between the vestibular nuclei and the nodules uvula. Here it's just drawn as sort of a 2-way arrow, but think of it as a loop. And the idea is if you go in and, whoops. If you go in and lesion this structure, you actually make the time constant in the vestibular nuclei go down from about 20 milliseconds to about down to 5. Or sorry, seconds, not milliseconds. I keep saying milliseconds. It's seconds. So this is a very important thing to help us retain our sort of sense of movement through space that compensates for the biomechanics of the end organ, right? And it's basically through this sort of reciprocal feedback with this part of the cerebellum that the time constant is lengthened. It's a kind of integration. I won't have time because I think I'm over time, but there's another very interesting part of the cerebellum that's involved in generating vestibular ocular reflex adaptation. So you can look at the next couple slides if you're personally interested. I won't test you on this. But a lot of us wear glasses or contacts. When we put glasses on or take them off, basically these are magnifying lenses or minimizing lenses, right? So whenever you do that, when you move your head movement, you're going to want a different eye movement, right? Because you have to adjust it for the movement of the visual world. If you're wearing lenses, it'll be different than if you're not. So there's basically a circuit within the flocculus, which is another part of the vestibular cerebellum. These neurons are actually hooked up with the PVP neurons that generate the VOR, and they're there to help keep the VOR pathway in calibration. So just like I talked about the RFN, which is actually connected to the nodulus uvula, being involved in calibrating vestibular spinal pathways, you have a separate pathway that calibrates your VOR. So I'll leave it, and again, you can look at the next couple slides, but that's primarily what is in the slides that follow that would be, I guess, primarily relevant for understanding cerebellar function. And then the only other thing I'll mention is at the level of this cortex, you have multiple areas of cortex that have vestibularly sensitive neurons in them. And if you go and record from those areas, again, this is just for your own information, there's a lot of multisensory integration, and you get in this idea of how do you coordinate the reference frames of visual inputs and vestibular inputs so that you can come together with one unified representation. So that's where a lot of current work is focused on, this idea of reference frames between different modalities. So I'll stop there. I'll thank everybody for their attention. I think everyone knows for the homework that you guys get an extra week, right, to hand it all in. So that's a good thing. Okay, thank you.
Yes. For question 3.